Hello, my name is Daniel Haas. I'm a new faculty member in the Department of Ophthalmology, so I'm here to talk about what my lab is going to be focusing on. And I have sort of a couple slides. This slide is dedicated to the very high level goals of the lab. The next slide is dedicated to the technologies that we're going to use to approach those goals. And then the last slide is really dedicated to the kinds of research questions in a much more narrow sense that we'll be asking. This is maybe more along the lines of ideally what we want to know in the very long term. And so, goal number one is understanding the eye at a sort of mathematical level, at a mathematical model, you might say, of the eye. I want to be able to contribute to the generation of such a model. And there are many people who are doing it, who are doing variations on this theme. And we have faculty in the department who are creating digital twins. And I think that's an essential component of this as well. Uh, when I talk about generating a model I want to talk about it from a metabolic perspective. My specialty is in understanding how energy is used and what energetic fuels are used by tissues of the eye. And so because energy kind of sets a maximum in terms of what a tissue can do, then this, I think, will be able to sort of bound future sort of physics-based model eyes. Goal number two, again, very high level, is just to understand disease from a different perspective, from a metabolic perspective. And you'll find in the scientific literature about, let's say, glaucoma, age-related macular degeneration, and there are several disorders where this occurs that there's some kind of metabolic dysfunction. But the numbers of reactions are actually probed to determine this sort of so-called metabolic dysfunction are actually pretty narrow. And so with this goal, I'm really hoping to broaden our understanding of disease by expanding the number of reactions that we can probe that might be changed in disease and understand how they're different and how they interact with these sort of known pathways that are altered in disease. So we can really figure out what is causal and what is a consequence of some other change. And then goal number three is improving resilience to degeneration. And so I kind of I have this little metabolic map here in the middle just to indicate my interest in metabolism, how everyone around me should be memorizing this map. But this picture on the right side with a building out, this is formerly this Sears Tower. Um, this is just to show kind of my idea of how we can improve, how we can preserve the retina from degeneration. So it might be that in disease, the lights go out, sort of metaphorically, because, cells start dying. But if we can deliver power when the lights go out, it might be that we can save a lot of cells. So if we pretend that these are rods and cones, for example, these buildings, then maybe if we can improve metabolism in some very specific way, we can preserve the retina, in particular photoreceptors or even retinal ganglion cells from dying by providing a new source of power. And so those are sort of the high level goals. Again, trying to protect from degeneration, understand disease, and truly have kind of a model of the eye that we can really kind of program into a computer and understand what's going on under any sort of future perturbation. And so how are we going to approach these kinds of goals? I think the easiest explanation for these approaches are that I'm a sort of specialist in mass spectrometry. So I've spent a lot of my postdoctoral career focusing on both developing methods and probing metabolism using what are known as stable isotope tracers. And these tracers can be something like glucose that you would normally get in your diet. And it's just like a carbohydrate, for example. But the carbons on that glucose, normally each carbon weighs 12 mass units. And the glucose that we would use in these experiments to understand metabolism under normal situations and in disease, they would be slightly heavier than normal carbons in glucose. They'd have an extra neutron, making them 13C, 13 just referring to their atomic weight. And so each of these carbons in glucose is slightly heavier, And we can deliver glucose to a mouse, for example, by infusion or cultured cells as well. And we can track where that glucose goes and how quickly it goes to its destination. And we track basically how those carbons are shifting from glucose in glycolysis to pyruvate, and then in mitochondria from pyruvate to many different sort of intermediates. And of course, glucose interacts with many different metabolic pathways, so we're not restricted to understanding glycolysis or the TCA cycle. And we can use mass spectrometry, which is sufficiently sensitive to detect a difference between a molecule that carries a heavy carbon and a molecule that carries a light carbon. And we can also detect what molecule it is that carries those heavy or light carbons. And so, these are actual mice here. They've been sort of given this surgical catheter, and the catheter's on their back. They have actually these look like rats, but the point is that the catheter is a system through which you can deliver these isotopically labeled molecules, and we're not restricted to glucose. Um, it just so happens that glucose is fundamental to many different forms of metabolism. And so, we can deliver these metabolites, track where they go, and that will allow us to understand sort of a lot of the quantitative parameters we need to build a model I, as well as how those parameters are altered in disease. Um, and we also have some slightly more updated technologies. These infusion systems have existed particularly for radioactive tracers. These 13C molecules are not radioactive, but they have existed a long time for radioactive isotope tracers. And they've been used for a little bit of time with these stable isotopes as well. But what's really new is the ability now to image the distribution of isotopically labeled metabolites in situ. So we can, infuse a mouse with 13C glucose, for example, and take a tissue out and slice it like we would for histology, though we have to adapt the experiment a bit. And we can put that tissue slice on a mass spectrometer that basically takes a mass spectrum at different pixels along the tissue to create an image, a map of how the metabolite is being used over space. And so these are sort of example images of, I think this is a kidney, and the distribution of how glucose is used in this kidney to make UDP glucose is different than the distribution of how glycerol is used to make UDP glucose. And this sort of reflects gluconeogenesis in this latter part. And so I think the general message is that if we take this technology to the retina, which hasn't really been done, to the extent that it could be, especially for isotopic tracing, then we might be able to really figure out reactions and how they're happening at a cellular level, not just in a sort of tissue homogenate, but really what's distinguishing photoreceptors, bipolar cells, and ganglion cells metabolically. And that will really help in generating a sort of a model and an understanding of how different cells are affected in disease. And so, I kind of talked about high level goals, but I think it's really important to be specific because I think we have a lot of attainable sort of bite-sized goals, let's say. They're not exactly bite-sized, but they're closer. So I wanted to go over a couple questions that these might be. Um, One of these, the first one, and I'm actually funded to answer this question, is really to understand whether metabolic alterations in the retina or in the RPE can precede degeneration in models of photoreceptor degeneration, inherited photoreceptor degeneration. And so that's a very reachable goal. And slightly less reachable, but I think still doable, would be a couple questions in age-related macular degeneration. One of these is very sort of oriented towards basic biology, but because AMD involves a buildup of lipid in the eye, we should really have a better understanding than we do of how lipid gets in and out of the eye. What molecules is it trafficked on? And when lipid gets in the eye in a certain form, how quickly does it convert to a different form of lipid? These are very tenable experiments to do with isotopic tracers. Goal number 3 is more related to the etiology of the disease, when lipid builds up in the eye and these things called drusen. The hypothesis is that that drusen blocks the movement of molecules that would nourish the retina and the RPE. So I think we can try to answer the question using mouse models and isotopic tracers of whether or not a lipid barrier is even a feasible idea. And then this last one, I'm kind of involving a new disease here, Leber's hereditary optic neuropathy. And I think it's just a fascinating disorder where photoreceptors and retinal ganglion cells in the retina, they both have incredibly dense populations of mitochondria. But mutations in mitochondrial DNA that affects how the mitochondria work only seem to affect retinal ganglion cells. And so understanding why that is, and when these mutations happen, they cause Leber's hereditary optic neuropathy. So understanding why these mutations affect only one population of mitochondria, but not the other at the sort of disease level is a fascinating question and really could help patients. So yeah, that's kind of the overview of what my lab will be focusing on. And if you want to talk to me, if you have any ideas, if you've already answered my question and you'd like to tell me all about it, please reach out. I'm in Clinic B, room 5602, and this is my email. And this is Yutao, who's going to basically be doing all of my work for me. All right, thank you very much.